All right, welcome to NBDE Microbiology Review Series number 4. Okay, so if you haven't reviewed the number 1, number 2, and number 3, make sure you review them, okay? Because it's very critical that you understand the bacteria, okay, in general, the size, where the DNA is found, okay? And you need to understand the cell wall of bacteria, the peptidoglycan, and then you need to understand that the peptidoglycan is made out of the repeating the saccharide units, all right, of N-acetylmuramic acid and N-acetylglucosamine, all right. And you have to know the penicillin binding proteins which are found in the cell membrane of the bacteria. Okay, and these are targets of the antibiotics, the penicillin and beta lactams. And you have to understand the virulence factors of gram positives and gram negatives. The gram negatives, we discussed that there's the lipopolysaccharide, which is LPS. LPS is endotoxin, okay, which can um, enhance the host immunity, okay, or it causes a response to the host immunity. We also discussed the some of the virulence factors, okay, uh, of the gram positive, such as the capsule, okay, or the gram negatives. So the capsule is a slime layer, and it allows the bacteria to evade phagocytosis. Okay. All right. So let's move on. Make sure you review all those things. Those are the key high yield information that you need for exam. So review number 4. Let's discuss about bacteria reproduction. Okay. So reproduction occurs via asexual reproduction or binary fission. Okay. Asexual reproduction via binary fission. And so in this case, in the asexual reproduction, coiled, coiled plasmic membrane structure called the mesosome binds and separates the daughter chromosome during division. Okay, and sometimes they form spores. Bacteria form spores, especially the Clostridia. Okay, Clostridia form spores, and also the anthrax form spores. Those are spore forming bacteria, and sp- sp- spore formation. One exception to the fact that bacteria don't have organelles. Okay, so bacteria don't have organelles, but they form spores. All right. Now, spores. Let's discuss spores. Spores are reproductive structures that are formed by gram positive organisms. Okay. Gram positive organisms such as Bacillus, okay, and Anthracis. That's the one which forms anthrax. Clostridium, okay, Clostridium tetani, the one we could cause tetanus, and Coxiella, all right, Coxiella barnetti, which causes Q fever. All these form spores, okay. Spores consist of multi-layered wall surrounding a dehydrated. So spores are dehydrated cytoplasm and DNA. So dehydrated cytoplasm and DNA. Okay. Very key slide here. So you have to know, make sure you know the information on this slide. And spores are highly resistant to heat, UV, ultraviolet light, chemical, or drying. Okay. So, which is the reason spores are the gold standard, which are used to see if the autoclave machine works. You put in spores, and if those spores are no longer growing or they change color, then you know the autoclave is working. Okay, because the spores are really difficult to 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 destroy because they are resistant to heat, UV, chemical, or drying. The spores can survive for centuries, hundreds and hundreds of years, even under harsh conditions. Okay, which is the reason that spore forming bacteria are dangerous. Okay, so make sure you understand spores. How about biofilm? What's a biofilm? Okay, so bacterial adhesion promote formation of biofilms. Biofilms are they are masses of bacteria, okay, encased in large aggregates of extracellular matrix. So bacteria get together in a community. So a community of bacteria, different species of bacteria, they benefit each other. You have aerobes, you have anaerobes, and then in between you have obligate aerobes, okay, or facultative anaerobes, okay. So all those species they get together and they form a community. So now a community of different species of bacteria is called a column, okay. Quorum sensing is how biofilms work. Okay. So once one bacteria senses something else, it produces protective factors to protect the community, to protect the quorum. Okay. A community of different species of bacteria again is called a quorum. Okay. A biofilm is a type of quorum. A type of biofilm in dentistry is calculus. Calculus, okay. Plaque calculus is a biofilm. Okay, make sure you know that. So most bacteria live largely in biofilms rather than as free organisms. Okay, and the biofilm, the reason they live in communities because communities you protect each other. Okay, so versus when you live by yourself in an isolated area, you are very vulnerable to insecurities. Okay. However, when you're in a community, there's more protection than when you live alone. So that's what bacteria do. They live in communities called biofilms. Okay. And this is uh, key for survival because remember, it's very hard to kill bacteria in a biofilm. Okay. Biofilms are more resistant to antibiotics. Okay. Because they are ward of community. Okay, so biofilms are resistant to antibiotics. Now remember, one bacteria, one type of bacteria may not be as resistant to the antibiotics you are giving. However, the other bacteria they have mechanisms that causes resistance to that antibiotic. So they use that mechanism to expel or to neutralize the antibiotics. So even the other bacteria which don't have any defense mechanism against that antibiotic, they still grow. Okay, because they're being protected. All right. So you have to understand the biofilm. All right. And example of biofilm is dental plaque or calculus. Okay, so the genetic transfer of information, we have three types: transformation, conjugation, which is conjugation is bacterial sex, and then you have transduction. Okay, so let's discuss each individually. So, what is transformation? So, transformation is a process by which bacteria take up naked fragments. So, they take up what? Naked fragments of DNA. It can be in any environment. Okay, as long as the bacteria is there and there's some DNA, the bacteria just takes up that DNA and incorporates that DNA into their own genome, into their own DNA. That is transformation. Make sure you know the definition of transformation. Trust me, you'll get a question on this. Okay. And their recipient cells must be competent. So, whatever cells of bacteria that receive the fragments of DNA, those cells must be competent. They must be alive. Okay, for them to incorporate the other DNA into its own. All right. Incorporation occurs by recombination. Okay. All right. How about conjugation? Conjugation, also known as bacterial sex. All right. So, conjugation is the transfer of DNA via a conjugative F plasmid. Okay. Plasmids are circular DNAs. All right. It's a extra DNA of the bacteria. And this plasmid, okay, is transferred from the donor, all right, to the recipient. It's so similar to transferring of sperm to the egg, all right. That's same with bacteria. You have the DNA of one bacteria, okay. So you have this bacteria with the plasmid transferring, okay, the plasmid, all right, during cell to cell contact, and occurs only between closely related cells. So, closely related bacterial species are the ones which perform conjugation. All right, and that makes sense. Okay, and plasmid. So, the purpose of plasmid, you have to know what a plasmid is. Make sure you know before you go in this exam. A plasmid encodes and transfers through the F pillars. Okay, so you can say that this is the F pillars, and this plasmid is transferred through here and goes here. Okay, all right. The so conjugation. So the pillars, a pillars is a conduit for DNA to pass through. Okay, and plasmids often carry antibiotic resistance, which is the reason bacteria exchange plasmids, because these plasmids they carry antibiotic resistance. Okay, and plasmids may carry fragments of chromosomal DNA. Okay, all right. How about transduction? What is transduction? All right. So transduction is where the DNA is transferred between a donor and recipient cells by the use of bacteriophage. So bacteriophage is actually a virus for bacteria. Okay. So you use a virus, and that virus takes in whatever DNA you want to transfer, and then the virus infects the bacteria, and then once it infects the bacteria, it transfers that DNA to the bacteria. Okay, So the virus is transferring one the DNA from from one type of bacteria to the other type of bacteria. Okay, and the host DNA is inadvertently packaged into phage capsid. Okay, capsid. So the capsid. So that's what I'm saying that the virus attacks the bacteria, and then its DNA is incorporated into the virus, and the virus attacks another bacteria and transfers the its own DNA into the bacteria. Okay, so and then once the DNA is packaged into the to the phage to the virus capsid, is delivered to the another host bacteria. Okay, and then integration of the host DNA occurs. So make sure you know the definition of transduction, transformation, and conjugation. Okay, our next one is going to be NBD Microbiology Review Series 5. Okay, make sure you review the other series that um, we did before this one. Okay, if you have any questions, comment or send me an email. Okay, and you can also send me a request of a topic that you want me to go over. All right, good luck, study hard, and stay positive. Okay.